in the context of, say, we start with a GC base pair. Okay, and again, we'll do the same type of reaction where we have a deamination reaction, and that results in a G base pairing. And I'm drawing these lines in the context of the DNA strands. So what's going to happen to the daughter that contains the U when it gets replicated? What will a polymerase place opposite of U? A, right? Okay, so if we undergo replication, one of these strands with the U will have an A placed opposite it, and then the other daughter strand, what's going to happen here? Obviously, because we have a normal G, we're going to get a C inserted here, right? Okay, so that's fine. It's propagating its information as it normally should, right? So this one's okay. However, this one up here is not fine, and because now we've actually introduced a sequence change. So there's still damage here, but what we also have now is an accompanying that's accompanying this damage is a DNA sequence change, which is a mutagenesis. And then, so this is like the first round of replication. So the first round. Okay, so here's mutagenesis. And if you follow this through, this one here, through a second round of replication, if we replicate this thing again, we'll get another UA base pair in one of the parental daughter strands, and then we'll also get an AT base pair. In the other pair of strands. And what's the consequence of this? Well, now we're at a point where there's no way for a cell to know or determine that this AT base pair that we've now created is wrong, because it's just a normal base pair, right? And in these other instances, you know, there's still damage present, and it's always possible for damage to be repaired before replication occurs. But once you've gone through 2 rounds of replication, this one here is not a repair template. Okay, so it is not a repair template, because what is presented is a totally normal AT base pair. So it's a normal AT base pair. And the only thing is, it's just not the right base pair. So you've had a sequence change. So sequence change. So this is akin to a cell being completely blind to this change. So obviously, from a cellular perspective, if you have a damage event, you want if you want to repair it, ideally it would be before you do something to repair it, like having a means of removing the U, such that you have the proper template G present. So at least there's some hope that we could repair this deamination damage that's occurred to the C. Okay, so just to reiterate, if it's possible to repair, here, the damage here, to make it correct, so that you could have you know, something that's blank, and you could use the normal G template so that you could get the correct insertion of a C here. Okay, so it's still possible. Also, repair is, I guess, still possible here. But I hope you'll note that if you do remove the U here, the result of the repair event is still not going to be correct, because you'll have something blank that's opposite an A. And if you use this strand as a repair template, you're still going to get a mutagenic outcome. Okay, so basically it's pretty critical that you take care of the damage prior to replication so that you're not altering the repair template. But at any rate, in a general sense, I just want you to be able to sort of distinguish the damage, which is the alteration to the base itself, from the mutagenesis, which is the conversion of the damage to a different DNA sequence. Okay, and I think the last thing maybe that I want to say about this, of course, is that once you kind of get to this TA base pair, this is totally a heritable DNA sequence, right? So this mutational event is heritable, but the damage is not. So one way to think about it is that if you replicate, this UG base pair, the damage is not propagated by replication, so it's not heritable, but the mutagenic product is heritable because it's a normal base pair. Okay, next kind of spontaneous DNA damage that I want to talk about is oxidative damage. This was alluded to earlier when we mentioned reactive oxygen species and free radicals as sources of spontaneous DNA damage. So oxidative damage is, of course, a very predominant form of DNA damage because we respire, and in the course of respiration, there's generation of what are referred to as reactive oxygen species. And the idea is simply that in oxidative phosphorylation, I guess the game at the end of the day is that we're passing a bunch of electrons down the electron transport chain. So if you remember, in electron transport chain, we're passing electrons down this chain, okay? And what happens to these electrons once they get down to the end of the electron transport? They're passed to oxygen, right? Okay. And then, we produce the water, so they go on to the water. However, this process is not perfect. Not all electrons end up on the terminal electron acceptor like they're supposed to. And I guess depending on the age of your mitochondria, about 0.2 to 2% of these electrons actually can leak directly to oxygen. So can leak directly to oxygen. And we get this. And that's the formation of superoxide anions. So superoxide anions are reactive for sure. They're not as reactive as hydroxyl radicals, which are probably the most strongest oxidants that end up getting produced by reactive oxygen species, by products of respiration. So these superoxide anions are converted to hydrogen peroxide, and typically this is done enzymatically. so enzymatically. There's an enzyme called superoxide dismutase, so SOD, that catalyzes this reaction. And what's sort of interesting about hydrogen peroxide is it is not a particularly strong oxidant, and in fact the conversion of superoxide anions to hydrogen peroxide is kind of a detoxifying event. Nonetheless, the issue with hydrogen peroxide is that it can cross membrane barriers pretty readily. Okay, so it can cross membranes readily. So where the superoxide anion might be somewhat restricted to the mitochondria, where we really don't care all that much because we like our nuclear DNA more than we like our mitochondrial DNA, hydrogen peroxide is a little more freely diffusible. Hydrogen peroxide in turn can be reduced, and it can be fully reduced to water. Okay, so it can be reduced fully to water, or it can be partially reduced to produce hydroxyl radicals, or it can be partially reduced to produce, again, these hydroxyl radicals. And so partial reduction we'll get this hydroxyl radical. And hydroxyl radicals are extremely strong oxidants. So these are strong oxidants. And strong oxidants can cause a large number of different kinds of DNA-based lesions. There are, in fact, on the order of 100 different kinds of DNA lesions that can be caused by reactive oxygen species. So approximately over 100 different lesions that can be caused by reactive oxygen species. These can encompass almost any kind of damage that you can consider. So damage to the bases, damage to the sugars, breaks the DNA. They can catalyze cross-links between proteins and DNA. So there's a lot of damage that can result because of this. In the interest of time, we will focus on just one. And so the one that I want to discuss is an oxidized form of guanine, which is shown below, which is 8-oxo-G. And this is kind of an interesting form, I think. So 8-oxo-G is one of the most abundant oxidative lesions, and as you'll see, as an abundant lesion, there's a considerable amount of cellular DNA repair capacity that is dedicated to fixing or removing 8-oxo-G from DNA. So the idea is pretty simple. If we have some kind of hydroxyl radical, so first, let me just write here, it's one of the most abundant oxidative lesions. Okay, so say we have a hydroxyl radical here. Okay, and we have a nice little position. See over here at position number 8. That's susceptible. We end up with, okay, so we go to that very susceptible position. We end up with an 8, if we have this carbon that's being attacked, and then now we have this oxygen or carbonyl group that's been inserted. And I want to point out at this stage, although obviously the chemistry in the base is altered, it's not super obvious how this change would affect the base pairing capacity, right? Because the base pairing action, I mean, it's all happening over here, right? If we think about how G pairs. And again, hydrogen bond donors and acceptor profile. If we look at 8-oxo-G, it's actually still the same, so it's unchanged. Okay. However, when you do go from guanine to 8-oxoguanine, 8-oxo-G is very, very, very highly mutagenic when it is present in DNA. Okay, so keeping in the theme with 8-oxo-G and how I left off by saying it is highly mutagenic when it is present in DNA. And so what I'm going to do now is explain to you how this actually works. If you recall, bases have a couple of configurations with respect to the sugar, and they're referred to as anti and syn. So we talked about this in the first section of the course. So which one is normally preferred? If you remember, it is the anti-conformation, right? And so this one here. And in the anti-conformation, as I explained a minute ago, 8-oxo-G presents exactly the same hydrogen bonding profile as G does. And so in the anti-conformation, 8-oxo-G will also base pair with C just fine. Okay, so in this conformation, you have the same H bonding profile and pairs with C. Okay, and so there would not result in any mutagenesis at all, of course. However, the presence of the 8-oxo-G introduces a problem. And the problem is simple, that it introduces a steric clash with the DNA backbone. And the steric clash is actually right here. So the steric clash with 8-oxo-G. And the result of this is that the 8-oxo-G actually does not prefer the anti-conformation. It actually now prefers the syn conformation. Okay, and now in the syn conformation, you of course don't have what you would call normal Watson and Crick base pairing with an incoming base. So this actually then becomes a Hookstein base pairing situation. And in this instance, the base pairing hydrogen bond donor and acceptor profile of the 8-oxo-G is quite different, and now it can base pair very readily with A. So we see the base pairing undergoing here and here. Okay, so in the syn conformation, the H bond profile is quite different. And it base pairs readily with A. Okay, and so I think the only thing I wanted to point out here is that in addition, and this is in contrast to G, what also facilitates this 8-oxo-G binding to A, base pairing with A, is that it has a new H bond donor at this position. Okay, so there is a new H bond donor in this position, and that position is N7. And this kind of sort of facilitates this base pairing with A when it's in the syn conformation. So again, if we sort of walk through the damage mutagenesis scenario, we start with a GC base pair. Okay, we go to an 8-oxo-G. And this is the damage. So the base has become altered. It has altered base pairing properties, but the damage itself is not heritable. When this becomes replicated, so replication occurs, there's the potential of 8-oxo G pairing with A. Okay, so it has a high frequency to do this, and this now becomes mutagenesis. And then if we undergo a second round of replication, we get our TA mutation, and this TA base pair is now appearing as a normal base pair, but of incorrect sequence, and this is entirely heritable. Okay. Okay, so next kind of damage is UV damage from Mr. Golden Sun. So this is one I'm pretty sure you're hopefully familiar with. And the idea here is that UV light can be absorbed by pyrimidine bases. Okay, so UV light absorbed by pyrimidine bases. Okay, and this can result in the opening of the ring, the carbon-carbon double bonds in those rings. So you get opening of the carbon-carbon double bonds. And if this happens, that ring opening really allows it to react with something. So reacts with something. And if it happens that there is a second pyrimidine in close proximity, as shown in this figure, where there's a T and a C, then there's two possible outcomes, both of which result from the reaction of this opened ring double bond with an adjacent pyrimidine. And the first is, and we'll look at it over here, is that you can get this sort of 4-carbon ring form, which is called the CPD, or a cyclobutane pyrimidine dimer. And this is the 4-carbon ring structure that's formed. And the second one over here is that rather than have the cyclic form where you have 2 bonds are formed between the adjacent pyrimidines, you actually just have a single bond that's formed. Okay, so just one here between the adjacent pyrimidine. And this is referred to as a 6,4 photoproduct. Or in other words, a 6,4 pp. So UV light absorption, ring opening, reaction with an adjacent pyrimidine has 2 possible outcomes. One is the cyclobutane pyrimidine dimer, where you can form 2 new bonds, and then the other is a 6,4 photoproduct, where there's only a single carbon bond between the 2. Alright, so let's delve into what are the consequences of this type of UV damage. Well, one sort of consequence, I guess, is geometric, and this is what's demonstrated here on the slide. So here is a sequence of DNA with a couple of T's adjacent to each other, and by virtue of exposure to UV light, which you've highlighted over here, this forms a cyclobutane thymine dimer, okay, and that's depicted in the lower part of this figure. And the consequence for this on the structure of the DNA is shown to the right. and that's shown in comparing figures C and D. So figure C, this is the structure without the dimer between the T and the T, and then figure D is the structure of the dimer between the T and the T. And I hope you can see that these things don't look anything like each other, and the reason for this I think is pretty easy to understand. So if we have a normal B deform DNA, if we look at this figure right here, and you have nicely stacked bases, how far apart are these two adjacent thymine bases? So what's the typical distance between here? So you remember back to, I think it was our second lecture or third, I'm not sure. This is about, or the first lecture actually. This is 3.4 angstroms. Okay, so now what's the length of a carbon-carbon single bond? Is it 3.4 angstroms? No, it's likely around 1.5 angstroms. So this is 1.5 angstroms, and that's typical length of a CC bond. So basically now what you've done is taken a molecule that's very comfortable structurally with the 3.4 angstrom spacing between adjacent bases, and you've constrained it to this new geometry where you only have 1.5 angstrom spacing between the two adjacent bases. And the result is this very dramatic kink in the structure, and we see it here. And of course, this makes quite a large disruption to the normal structure of the DNA helix. It also disrupts base pairing. You can see everything kind of looks a little weird down the axis. So if this is the normal one, this kind of looks very weird here. So there is this distortion that's occurring. And then perhaps more importantly, these UV lesions are physical impediments to the passage of polymerases. So both DNA and RNA polymerases are unable to readily bypass these types of lesions. However, as I mentioned previously, there are specialized translesion synthesis DNA polymerases, or TLSs, that can bypass different kinds of lesions, including A-base sites. There is a specialized TLS polymerase that can bypass UV damage, and what's sort of interesting about this is that the TLS polymerase for UV prefers to insert AA opposite UV damage. Okay, so the TLS polymerase for UV prefers to insert AA opposite UV damage. And this can occur opposite either a pyrimidine dimer or a 6,4 photoproduct. And what's sort of cool about this is that if you consider what this would look like in the context of a thymine dimer, so you might have something like this in one strand, and then the other strand has A's opposite it. You know, you expose it to UV, and then we get the generation of these thymine dimers in the one strand. Okay, and it's still base pairing with our AAs. And then we undergo replication. Okay, so the strand that has the thymine dimer in it is going to have the TLS polymerase actually insert a pair of A's opposite it. Well, that's great. And again, obviously the other strand is all fine and dandy. So in this instance, even though there was DNA damage, the TLS polymerase has inserted A's opposite it. So there's no mutagenesis, right? Because it inserted the correct bases. So the sequence is unchanged. So no mutagenesis. Sequence is unchanged. Is unchanged. Okay, so that's great. You might appreciate, however, that the TT is not the only combination of two pyrimidines that you could have, right? So the strategy is great some of the time, but what would happen if you started with a CC dimer? And then this strategy obviously fails, right? So if you had a CC dimer, so what's going to happen? So we can draw this here. And again, this would normally be base paired with a G. If we undergo a replication or TLS polymerase, we'll insert A's. Okay. So this TLS polymerase isn't looking so smart anymore, is it now? No, because it's put AA, and then of course with one more round of replication, this mutation then becomes fixed, right? Okay, and now that we started with a CC, you know, the base paired normally, so we have this one here. It's now been converted through this mutagenesis to a TT that's base paired with these A's. And this kind of mutagenesis, where you have a pair of C's going to a pair of T's, is actually the signature of UV mutagenesis. So this going to this is the signature of UV mutagenesis. So if I was to sequence my genome, and in particularly if I was to sequence the genome of, say, one of my skin cells, and I looked at a particular gene, and I know what the wild type sequence is in the gene from one of my skin cells, and then I see that there's some positions in the wild type sequence where there's C's, but in my skin cells they're T's, I can infer that the mutation arose because of UV exposure. And the mechanism by which it arose is exactly this, that there was almost certainly a pyrimidine dimer between the C's that was, you know, bypassed by a TLS polymerase, leading to the sequence mutagenesis occurring where CC is changed to a TT. Okay, so just to finish off the final thoughts of UV damage, I just wanted to show this slide, which contains two structural models. One that has a CPD on the left, and one containing a 6,4 photoproduct. And I hope that you can see in both cases the physical constraints imposed by carbon-carbon bonds between adjacent pyrimidines leads to this sort of distortion of the DNA structure and a kinking of the DNA helix. In addition to mucking up the base pairing across from the CPD, which you can also see, the bases on the opposite side sort of maintaining their normal base pairing stacking, which obviously is not compatible with them being in the same plane as the constrained bases for proper base pairing. And that will become important a little later when we look at how UV damage is repaired in a cellular context. The final form of damage I'm going to talk about is alkylation damage. So alkylating agents are among some of the most potent environmental carcinogens. Okay, so potent carcinogens, and again, they come from all kinds of sources, like burning tobacco is a good one, grilled food, moldy grain, moldy peanuts are good examples. So there's really a lot of fairly complex alkylating agents. The simplest alkylation, and the one I'm going to show you for simplicity's sake, is of course methylation. All right, so methylation. And this is true of alkylation in general. It can occur at a variety of different positions on bases, at both nitrogens and oxygens. Okay, so we can have methylation occurring on oxygens and nitrogens. And there are a number of different ways in which alkylations can alter the base pairing properties of bases that have become alkylated. So the example I'll show you is methylation of oxygen 6 of guanine to produce O6 methyl guanine, which is depicted here. Okay, so it's just a very simple example. And the important thing to note really is what effect this has on the hydrogen bond properties of the guanine. So normally the H bond action here is typically what? An acceptor or a donor. So if we look at this oxygen, that's an acceptor, right? In this position. What about the one over here at this position? This is a donor, right? We have an amine group. And if we look at O6 methyl G, these have been changed. Both of these positions have been changed due to the methylation.